Hello, good morning, everyone. Hello, good morning, everyone online. You are most welcome to our second day of our second session of this third joint Global Foot and Mouth Disease Research Alliance and the EuFMD Regional Workshop in Africa. Yesterday, we had a very good introduction and explained the purpose of the meeting. We aimed to bring researchers, policymakers, and implementers interested and involved in foot and mouth disease control to share their knowledge, experience, and ideas, and also to identify gaps, and also share the ideas on how to address them. Again, I come back to speak to you. Yesterday, we had very many attendants from all over the world. There are 180. I hope now also the number will increase, such that we can share our knowledge globally. We had 5 presenters yesterday. That is Dr. Abraham Sangula, Dr. Donald King, Dr. Badi Marudi, Dr. Mbakachi, and also David McKay. They gave very good, interesting presentations. That was research in Africa to support vaccine quality by Abraham. Importance of independent vaccine quality control by Donald King, the private public partnerships of vaccines of FMD by Dr. Badi, and also availability and use of FMD vaccines and the role of the stakeholders. Of course, we ended with another good presentation, the pre-qualification system for FMD vaccines in support to assured emergency supplies and options. Colleagues and members, I hope all of us really attended yesterday. I have got this one to share with you, such that we are on the same page. Of course, this is the slide that I presented yesterday, but it's a small recap. So this is our Frank Norbert Mwine is once my name. Again, the current president of the Global Foot and Mouth Disease Research Alliance from the College of Veterinary Medicine, Animal Resources and Biosecurity in Makerere University here at Kampala, Uganda. Of course, we very much know our agenda for these 2 organizations that are working hand in hand. Such as
Frank, sorry, can you switch off your camera, please? You're breaking up a lot. Hello, everybody. Really sorry. I think we've lost Frank completely now. So apologies for that. And I hope you don't mind if we then just continue with the program. I'm quite sure we'll find Frank again, and Frank will find us along the way. So I'm just going to introduce myself. So I'm Wilna Vosloo. I'm part of the Global Foot and Mouth Disease Research Alliance Executive Committee, and I'm based at the Australian Centre for Disease Preparedness in Australia. So just want to say hello to everyone, and I'm really happy to have you all online with us again tonight. So it is my absolute pleasure to introduce you to the chair of tonight, who will take over from me and take you through the program. So Dr. Chris Ayebazipwe is a veterinarian working with the Food and Agricultural Organization of the United Nations in Uganda. He holds a PhD on foot and mouth disease from the Makerere University and the Danish Technical University. Previously, Chris worked with the Ministry of Agriculture, Animal Industry and Fisheries, in Kiruhura and Mbabara district local governments, focusing on foot-and-mouth disease research, control, and diagnostics. Chris has greatly contributed to the design and implementation of over 20 projects and to over 50 scientific publications. He supports the agricultural development and One Health at various levels in Uganda. So, Chris, I'm really happy to just hand over to you. Thank you very much.
Thanks so much, Wilna, and good morning, members. Welcome to this second day of the GFRA stroke EuFMD meeting. We have a number of presenters today, including David Hizibolo, George Omondi, Sylvia Baluka, Lesenyo Motapele, and Jones. So at this juncture, allow me to introduce David Hizibolo, who's David is a veterinarian located in Nigeria and obtained his PhD in 2016 from the Faculty of Veterinary Medicine, Ahmadu Bello University, Nigeria. He's currently based at the Infectious and Transboundary Animal Disease Department, Foot and Mouth Disease Virus Research Laboratory, National Veterinary Research Institute, Vom, Nigeria, where he is responsible for diagnosis, research, and surveillance of various studies. He's also a member of the Nigerian Veterinary Medical Association, Nigeria, and the Nigeria Biological Safety Association. Welcome, David, as he gives his talk this morning. Thank you.
Good morning, everyone. I'd like to thank the organizers for having me, and also greetings to other speakers and facilitators in this workshop. My talk is on foot and mouth disease surveillance in endemic areas. Disease surveillance is an important epidemiological tool used to monitor the health of a population. The goals of an effective disease surveillance program are mainly to describe the current burden and epidemiology of the disease, to monitor current trends, and to identify outbreaks and emerging strengths. FMD surveillance should be a continuous scrutiny of occurrence of the disease to enable prompt intervention for the control of the disease. The African and Asian regions of the world are recognized as endemic areas for FMD. Serotypes O, A, SAT1, SAT2, and SAT3 are the common FMD serotypes circulating in the countries of the African region. Cattle has been identified as the main FMD affected species during outbreaks. Is FMD underreported in endemic areas? There is no doubt that FMD surveillance in endemic areas of Africa is quite weak and grossly inadequate, resulting in underreporting of outbreaks and masking the true prevalence of the disease. Some reasons have been adduced for underreporting, which ranges from the inability to detect or report as a result of lack of awareness, logistic difficulties, lack of diagnostic capability, or the lack of communication, to the unwillingness to report due to lack of compensation and lack of feedback or response. FAO and OIE launched the Progressive Control Pathway for FMD in 2012 with the goal of minimizing the impact of FMD in endemic countries. The initial measures outlined in this strategy include early detection and alert systems and the implementation of successful surveillance. According to the OIE guidelines for animal disease control, the component of any surveillance program following an outbreak report must include data and sample collection, laboratory investigation, data analysis and interpretation, dissemination of information, and of course, action or response. Certain factors that need to be considered in setting the goals of FMD surveillance in endemic areas include biological factors like affected species, distribution and density of susceptible species, wildlife reservoirs, modes of virus spread, and also the availability of technical tools like diagnostic tests, trained personnel to control measures that involve movement control, isolation and quarantine, cleaning and disinfection, vaccination, to socioeconomic considerations that include cost and benefit of interventions, availability of resources, logistics, structure of livestock production systems, political will, and institutional arrangement. Assessment of these factors should guide the strategic planning of an effective surveillance program. In Africa, the livestock husbandry systems varies from region to region, mainly due to differences in the availability of grazing and watering points. Basically, it consists of pastoral nomadism, transhumance, and sedentary husbandry systems which are characterized by unrestricted animal movements, communal grazing and watering points, frequent intermingling of transhumance herd with sedentary herd, and also cattle being grazed and housed with small ruminants. FMD transmission in endemic areas is characterized by frequent direct and indirect animal contacts. The type and nature of the animal husbandry systems in Africa provides the epidemiological link for easy spread of the virus. I would like to share our experience on FMD surveillance in Nigeria from a recent EuFMD supported applied research project on the role of small ruminant in the epidemiology of foot and mouth disease. This is actually a collaboration between the National Veterinary Research Institute in Nigeria and the Pirbright Institute in the United Kingdom. Considering the paucity of funds available, we adopted a model which involves, first, selection or identifying high-risk areas. Then we conducted sensitization programs targeting livestock farmers, private veterinarians, livestock officers, and the community animal health workers. We conducted trainings for para-veterinarians and the community animal health workers in the target areas that included clinical science recognition, sample collection, sample shipment to the laboratory in the framework of biosafety and biosecurity, and also in the use of mobile app for data collection and outbreak reporting. We also provided sample collection kits to train field volunteers, and we ensured prompt response to outbreak reports. The integration of this model enhanced our surveillance approach. The surveillance approach used in the identified high-risk areas included passive and active surveillance. In the passive surveillance component, we investigated reported field outbreak, and we collected vesicular or epithelial tissue samples and data from affected animals. We also collected environmental samples from affected premises. In the active surveillance approach, we selected 2 local government areas within the high-risk area. In each selected local government area, we identified 1 livestock market, 1 transhuman site, and 5 sedentary households. We visited these premises once in a month for a period of 7 months. In each visit, animal and premise data were collected using a standardized questionnaire. Samples were collected that included oral swabs, blood and tissue samples were available from cattle, sheep and goats, and between 5 to 10 environmental samples were also collected. In the preliminary results obtained, 3 serotypes of FMD virus were detected in cattle across the local government areas in the identified high-risk area. Serotype A was detected in goats, and also serotype O was detected in sheep. Majority of the small ruminants in the same herd as the acutely infected cattle showed no sign or lesion of FMD. Cattle seems to be infecting the small ruminants within same herd. This is the first isolation of FMD virus from small ruminants in Nigeria. The map shows the spatial distribution of FMD serotypes in the identified high-risk area. Serotypes SAT2, O, and A were detected in the eastern part of the area, serotype O and A in the central part, in the western part, and also in the southern part of the identified high-risk area. Corresponding serum and oral swab samples collected from small ruminants associated with infected cattle herd were tested for foot and mouth disease non-structural protein and genomic RNA by ELISA and real-time PCR respectively. 70% of the small ruminants tested positive for FMD-NSP, while viral RNA was detected in 30% of the oral swab samples tested. Interestingly, FMD RNA was detected in environmental samples such as header sticks, boots, hard floor surfaces, pegs and ropes as part of the active surveillance, and also from fences, feeding troughs, transport vehicle, pegs and ropes during the passive surveillance. This confirms that formites plays a potential role in the spread of FMD in endemic areas. This depicts the zero to poor biosecurity measures on farms. Also, this study supports the fact that integrating studies for different transboundary animal diseases can optimize resource allocation. PPR viral RNA and Capripus viral DNA were detected from the same environmental samples collected during the active and passive surveillance for FMD study. This aligns with EuFMD proposal, which is to develop integrated programs for FMD and similar transboundary animal diseases. These viruses, the FMD virus, PPR virus, and Capripus virus, were detected more in the livestock markets across the selected local government areas in the high-risk area, also suggesting the poor sanitary condition of these livestock markets. In conclusion, Our surveillance approach was greatly enhanced with the integration of the simple model highlighted. Extending this approach to other areas of the country is likely to improve the generation of information on disease situation in Nigeria. To address the knowledge gap on the status of FMD and other transboundary animal diseases in endemic areas of Africa, political will and institutional structures, amongst other things, need to be reconsidered. No global FMD surveillance will be possible without a strong local surveillance. Therefore, improving surveillance systems in endemic areas, early detection and notification of outbreaks are key to understanding the burden and epidemiology of the disease, monitoring current trends, and identifying new strains of the virus. Thank you for listening.
Thanks so much, David, and very much appreciated. The presentation is very informative. Questions will be received in the question and answer window. I think at this juncture, let's move on to the next presenter, who is none other than Sylvia Baluka. Sylvia is just a moment. Yeah, Sylvia Baluka is a veterinarian with over 24 working experience in vet services, and yeah, she has experience in the vet service delivery, research, academia. Her work has focused on transboundary animal diseases such as foot and mouth disease, as well as foodborne diseases. She has worked with several organizations, including public and private sectors, development partners, and non-government organizations. Sylvia has worked and or collaborated with various stakeholders in livestock sector, including veterinarians, veterinary professionals, veterinary services at national and district local government levels, farmers organizations, and cooperatives. She's the immediate past president of Uganda Veterinary Association. Sylvia is currently working as a senior lecturer at the College of Veterinary Medicine, Animal Resources and Biosecurity, Makerere University, and a consultant with EuFMD on the project, as a project coordinator for Uganda for the Sustainable Business Animal Health Service provision through training of veterinary professionals. At this moment, allow me to welcome Madame Sylvia to give her talk. Thank you.
Thank you so much, Dr. Chris Aivaziwe, the chair of this session. Thank you, all the organizers, for giving me an opportunity to make this presentation. And I appreciate you all for giving me audience to share some summary findings, and also to share the goal of this project, which is being implemented as a public-private partnership. Our presentation is veterinary paraprofessionals' competencies and skills for an improved surveillance. As you've heard, the project is sustainable business in animal health service provision through training for veterinary paraprofessionals. I would like to acknowledge the contributions of all the project members who have contributed towards preparing this presentation. Here is an outline of the presentation. We'll give you the project overview and highlight the relevance of veterinary paraprofessionals, that we are going to abbreviate as VPPs throughout the presentation. We'll share with you summary findings on the role of VPPs in the control of foot and mouth disease and other similar transbound animal diseases that we'll abbreviate as FAST throughout the presentation. We'll also share with you the Ugandan case study highlighting the role of VPPs in disease surveillance. And then we will also share with you a brief on the VPP project competence framework that has been developed by the project. In this slide, we're just giving you the general overview of our project. And the general overview of this project, here is the broad goal: to increase agricultural productivity for smallholder farmers. And we will do that by improving the VPP capacities to deliver essential animal health services in key geographies to help reduce animal mortality and increase the productivity of livestock assets, particularly for the smallholder livestock farmers. The project is being implemented in 3 countries, Uganda, Nigeria, and South Africa. This is a 3-year project running from 2021 to 2023. It is being implemented as a public-private partnership involving the EuFMD, FAO, Health for Animals, and a World Veterinary Association. In addition to the overall goal of the project, we would like to share with you the key pillars upon which this project is anchored. The first pillar is competence-based training. This will be done via or using blended learning approaches. And the training is addressing both technical skills, such as in preventive animal health care, as well as the business skills and soft skills. The second pillar is focusing on certification. All the VPPs who will be selected and trained under this project will receive certification by the Veterinary Statutory Body, which is the Uganda Veterinary Board in the case of Uganda. The third pillar of this project is providing support to VPP business environment. And this will be achieved through strengthening the VPP linkages with farmers, the VPP linkages with veterinarians and veterinary medicine suppliers, and also by establishing or strengthening the public-private multi-stakeholder platform. We would also like, in this presentation, would like to answer the question of why should we focus on VPPs or veterinary professionals? We all note that livestock are critical to the livelihoods of smallholder farmers. And there is a growing demand for animal-based proteins and products. And this directly implies increased demand for animal health services. And as a result, there is a gap between the supply and demand of primary animal health services, more so in the remote areas. And this is due to limited presence of veterinarians, both public and in private sector. Now, this gap is often filled by the veterinary professionals. And that's why veterinary professionals are very core, and this project is prioritizing them so as to bridge that gap more effectively. Here, we would like to highlight the numbers of figures of veterinarians and veterinary professionals in Africa. We would like to pay more attention on Uganda, where this red arrow is pointing. Looking at all the other countries in Africa, you realize that Uganda, we are very low on both numbers for veterinarians as well as VPPs. While the blue bar, the number of veterinarians, the orange bar are the VPPs, the yellow dot are the tropical livestock units. Now you realize that the number of VPPs and veterinarians combined is still much lower than the tropical livestock units. And therefore we cannot get the appropriate ratios for us to achieve effective animal health service delivery. And also this graph gives an impression that the numbers of veterinarians and VPPs are almost equal, but in reality, we know that the number of veterinarians is much lower than the VPPs. And in any area that you go, especially in the remote areas, we are likely to find more VPPs than we are likely to find veterinarians across the country in Uganda. Here, I would like us to look at these three maps. The first map is highlighting the number of VPPs for the different African countries. The second map is showing the number of tropical livestock units across different African countries, while the third map is showing us the number of veterinarians per African country. Now, Uganda is this small country next to Kenya and above Tanzania and Rwanda, Burundi. So you realize that if we look at this picture, this color for the number of veterinarians, and we look at the color for the livestock, we realize, then look at the color here, from the key, which is showing the number of VPPs. Okay, these are the number of VPPs, and we have the tropical livestock units, and we have the number of veterinarians. This map was presenting the same picture like the graph that we had before. So we can't overemphasize that actually Uganda, we are still very low in terms of manpower, in terms of the human resources that are involved in animal health services. Now, this slide is giving us a summary of findings that were established by a survey that was conducted by the EuFMD at the beginning of this year. And this graph is highlighting the VPP roles that were identified by the survey. The roles include disease reporting, vaccination, exercise provision, sample collection and diagnostics, disinfection, disease reporting, animal movement control, and then we have animal disease control campaigns, and then culling. Culling, of course, especially in the cases of the TAD, such as foot and mouth disease, especially where there is a possibility for compensation, will be an important tool besides disinfection. The next, I would like to look at the Ugandan case study and the roles of the VPPs. We know them in Uganda. VPP roles in Uganda include the following: general awareness raising, disease prevention measures. Awareness raising and extension messages to farmers in regards to control of animal diseases is most often done by the VPPs. And also VPPs are involved in vaccination campaigns. VPPs are involved in mobilization of farmers and communities, and they also participate in administration of the vaccines. Many times they constitute a big number of the team members in these vaccination teams, of course, under the leadership of the district veterinary officers. VPPs also participate in providing advisory services to the farmers on animal health and production. The next role that we can look at that the VPPs play in Uganda, we have VPPs playing a role in disease surveillance. Here, VPPs communicate or convey information from farmers to district veterinary officers and vice versa. VPPs are engaged in routine meat inspection, and during meat inspection, during the post-mortem examination, many times they detect diseases based on the lesions, and they convey those reports to the district veterinary offices. Now, these reports take forms of, they could be in verbal, written, or electronic forms. VPPs are key stakeholders in animal disease detection, given their close interactions with the farmers. VPPs are usually part of the sample collection teams. They are the ones who lead the laboratory teams, and together with the epidemiology teams from the ministry to the farmers for purposes of routine sample collection, as well as in response to animal disease outbreaks, such as in the case of foot and mouth disease, where we have the frequency of outbreaks here is quite high. VPPs also play a role in communicating information from the district of veterinary officers to the farmers. Once a disease outbreak is identified, they're the ones who tell the farmers what to do, even when the quarantine measures are being enforced. They carry out sensitization and awareness raising as part of actions that are recommended following data collection and analysis during disease surveillance. VPPs play a major role in mobilizing the farmers or communities for different activities and for particular responses to an outbreak. For instance, when there is preventive vaccination campaigns have been organized after detecting FMD cases. It will be the VPPs that actually go out to the communities and they prepare and get the communities ready for these vaccination programs. VPPs also use delegated authority from the district veterinary officers to issue movement permits in some cases, especially in some districts where we have no veterinarian or we have one veterinarian who is seated in the office as a district veterinary officer. And in so doing, VPPs help to enforce movement control using the animal movement permit system. VPPs also manage and control the livestock markets using the delegated authority from the district veterinary officer. And in this project, we have worked together with different stakeholders, including the VPPs, including the veterinarians, including veterinary service representatives, drug regulators, and other key stakeholders, even farmers representatives. We have worked together under this VPP project and developed a VPP competence framework. And this framework has identified 13 core competencies that are very important for animal disease surveillance. And I would like to pull out five core competencies from this 15, which are linked directly to support the effective animal disease surveillance. Here we have herd and flock health and management. We have biosafety, biosecurity, and occupational health and safety. We have disease prevention and control, specimen collection, sampling and diagnostics. Then we have communication. Communication, which is like the bloodstream of a surveillance system. VPPs are very good in this aspect of communicating and bringing the different stakeholders together during the disease surveillance and also in response to animal disease outbreaks. Now, we would like to share with you a small example of how the VPP competence framework, how it can be applied. We are giving you on this slide an example of a VPP called Anna, who had already graduated with probably a diploma in animal health and production. So she had some knowledge already of disease surveillance, but for her to improve her competence in animal disease surveillance, she needed to do continuing professional development courses and guided by the VPP competence framework. So based on her earlier certification, knowledge, and experience, Anna realized that she's at the intermediate level of the framework, and yet she would like to achieve the advanced level. For Anna to achieve this advanced level, according to the competence framework, she needed to attend program C to be able to achieve additional competencies to help Anna to be able to participate effectively in disease control and surveillance. So this is how the competence framework can be applied. Different VPPs at different levels, and the competence framework has 3 levels. It has a beginner's level, intermediate level, and advanced level. We do believe that the VPP competence framework that the project has developed is adoptable and adaptable and can be used by other countries besides the 3 project countries, and even has utility beyond the life of the project, beyond the 3 years, which makes up the life of the project. By way of conclusion, we would like to restate that VPPs play a key role in disease surveillance and control. Given the close proximity of VPPs to farmers, it makes them a very important asset for community mobilization, communication of disease control measures, and ability to link veterinarians with farmers. The VPP competence framework, as I said earlier, can be used to identify competencies that are needed for improved animal disease surveillance and disease control. And the VPP framework, the way it is being developed, can be utilized by a single VPP to be able to match their own competence and therefore be able to identify their needs. And then they can be able to access the trainings under the continuing professional development or CPD programs for the different countries, of course, guided by the veterinary statutory bodies. And on a personal note, I would like to implore all of us and all the key stakeholders who are gathered here on this global foot and mouth disease research and alliance platform to prioritize the issues of veterinary paraprofessionals. Veterinary paraprofessionals comprise the weak link in animal health service delivery. And there are so many interventions we have undertaken with little success. And I think it's a high time that we address this weak link. And that's why I believe this project is timely, and with all the support of the stakeholders, we do believe this project will have an impact in strengthening veterinary paraprofessionals. And we hope that beyond this project, there will be other efforts, there will be other interventions, and there will be more resources that can be committed to this cause as we continue to prioritize the issues of veterinary paraprofessionals. I do acknowledge the contributions of the team from the EuFMD, FAO, comprising of Fabrizio, Leah, Holly, Emma, besides myself, who have participated in preparing this presentation and who are part of this project. I also acknowledge the other project implementing partners, who include the World Veterinary Association, Health for Animals, and of course, I would like to thank you all for your kind attention and audience, and to the organizers, I thank you once again for the opportunity to be able to make a presentation at this important global meeting. Thank you.
Thank you very much, Sylvia. Very good. It's a very interesting presentation on veterinary paraprofessionals, and we look forward to seeing how it intersects with surveillance and also the broader aspects of FMD and MST. Yeah, at this juncture, allow me to invite the next presenter, who is called George Omondi. George Omondi is a wildlife veterinarian and epidemiologist, and currently a research associate at the College of Veterinary Medicine at the University of Minnesota. His research interests span various facets of livestock-wildlife-human interface, where he applies novel ecological and epidemiological analytical tools to elucidate on the dynamics of pathogen spread with a view to answer questions on evolution, transmission, surveillance approaches, and control of infectious diseases. He has over 12 years experience, having worked as a wildlife veterinarian at Kenya Wildlife Service and all Pejeta Conservancy in Kenya. He recently completed his PhD in veterinary epidemiology at the University of Minnesota, and currently leading field and laboratory research efforts on foot and mouth disease and bovine tuberculosis in Kenya, and validation and optimization of tuberculosis diagnostic tests in great ape sanctuaries in 10 African countries. George, you are welcome. The floor is yours. Thank you.
Thank you. Hi, Sylvia. Could you allow me to share my screen?
Sylvia, kind and share.
Thank you. So I'll be sharing on research needs for improved surveillance in Africa, and I tried to take a slightly different route that I hope will be informative to this group. Thank you all for listening to me, and thank you for the invite. And that's been really nice presentation.
George, kindly put in full screen mode. Thank you.
Oh, sorry. Is that okay? So there has been really nice presentations between yesterday and today, looking at the different aspects of it that all actually do comprise of the research needs. And so I'll just basically start kind of putting across, excuse me, by putting across kind of the ecology and epidemiology of FMD in Africa. There's been a lot of scientific progress, especially in looking at kind of the drivers of the ecology and the epidemiology of this particular disease. This has been from looking at things like livestock systems, where we know the maintenance of the virus and the widespread nature of it in, for instance, pastoralist population or transhumant population. We know of the role of wildlife, especially the articulated role of buffaloes. We know of the issues about facets of geographical spread, even including kind of miniature transmission barriers in specific areas. And then we also know about the social ecological aspects, which include things like movement and the role of interaction between and within herds, but also between and within species. The other side of the epidemiology or the eco-epidemiology has been the transmission dynamics, where we talk about the direct versus indirect. And this is basically modes of transmission and how we can use those to basically try and evaluate both from what we've had from the previous presenters in terms of environmental transmission, environmental contamination, but also the role of common shared resources like common grazing grounds and watering sources. We think about the aspect of it in terms of viral extinction versus carrier status, and we think about this in the context of within and between farm and herd dynamics, where potentially within a farm you may have carrier animals, but also within a population you may have carrier animals, and that may lead to differences within it. Some of this has been elucidated by both statistical and mathematical modeling. And then the other aspect of things that have been really worked on is the diagnostic techniques, both from the individual to the farm or the herd level. And this also includes aspects of genomic technology, which includes phylogeography and phylodynamics that has allowed us to basically be able to track the pathogen, track its spread, but also be able to infer who transmitted to whom. These are important aspects of the ecology and the epidemiology because they allow us to actually think about the control mechanisms that we could use. Putting this together, we may think about things like surveillance system, where we have data collection gaps, especially in terms of in areas which have highly diverse populations like East Africa. There's a lot of work that has been done in Southern Africa, but in East Africa, as much as a lot of work has been done, there are still some gaps that I'm going to kind of lay that out. And this comes from thinking about targeted versus risk-based versus convenience versus random, and then potential alternative where we focus on specific areas that may yield better information that we can use. And this can be applied into control measures, including quarantine and vaccinations. The diagram showing on the left basically just kind of summarizes that in kind of a diagram manner. And so when you think about the knowledge gaps, which we also consider them as research needs here as well, I'm just going to expand a little bit on certain areas. I didn't want to go into all the areas in terms of listing down everything, but I discussed them in terms of knowledge gaps that are potentially out there. One of them is understanding transmission. Some of the knowledge gaps here are in kind of understanding how, why some serotypes tend to be more prevalent in certain areas, and why, for instance, you're losing certain, some of them, for instance, see, we're losing it. What does that mean, especially in the context of ecological adaptation or partition of what you can consider segregation in terms of why do you have this segregation in certain areas? And even within local geographical spread, you still have the segregation of some of these serotypes. The other aspect of it in terms of understanding transmission is thinking about the temporal and the spatial dynamics of the infection, especially in aspects like pastoralist, where we may start thinking about the potential for source and sinks. This can be interpreted as why would we have a lot of FMD and controlled FMD in some locations, especially knowing that in those particular locations, we do have certain factors that may contribute, but we do not have that being widespread across, or rather being homogeneous across. So there's some kind of heterogeneity in terms of the transmission. And this can also be thought about in terms of viral factors that promote persistence, emergence of new variants, and the transmission of those particular strains. The second aspect of it is thinking about how when some of the gaps in terms of understanding the host and the pathogen, host-pathogen interactions. Basically, what drives the host's preference and what kind of allows for selective advantages that allows for certain serotypes to exist or to occur in certain spaces. A good example is in 2016, we did sample Olpejata Conservancy, which is a contained environment, 90,000 acres. And despite the fact that there's mixed grazing of livestock and wildlife, we saw that the serotypes SAT1, SAT2 that we recovered from buffalo were not the same as serotypes A and O that we recovered from the livestock. And this was interesting in that even in terms of thinking about why would there be differences, despite the fact that in 2013, we allowed the FMD to basically run through the whole conservancy, partly because we do not have effective control measures at that particular moment in time, and our vaccine scenarios were basically geared towards animals at about 12 months of age, so a year. The other aspect of this host pathogen interaction is thinking about the detection of antigenic shifts, which basically means that the drivers in terms of immunity of the animal leads to potentially changes in the antigenic shift that leads to the emergence of new strains. How does this particular work, and what does that mean for vaccine efficacies? In terms of vaccine efficacies, some of the gaps are basically in trying to understand things like the role of viral epitopes. This is basically brought about because we depend a lot on sequence-based prediction of antigenicity and how we try to match the vaccines. But then we're starting to see this kind of emerging fields, not necessarily emerging, but this emerging knowledge base of viral epitopes. And I think thinking about how do we incorporate that in vaccine efficacies and vaccine matching may actually yield a better match for the vaccines vis-a-vis the field strains that we have. And here especially also is thinking about how do we predict vaccine performance in a highly diverse area that has multiple serotypes of FMD virus, or potentially even recombinations of these particular aspects, of this particular virus. And so the other aspect of it, the knowledge gap, and basically also consider a research thing before I kind of delve down a little bit into a narrow section that I prepared, is thinking about diagnostics and how do you differentiate from vaccine versus non-vaccine. And then thinking about the challenges we have, especially the lack of commercially available pen side versus field-based diagnostic tests. And then the application of molecular techniques, especially in tracking the virus. When we think about FMD persistence, this is where I basically kind of centered my talk on, rather than speak more broadly. I centered it on in terms of thinking about persistence in populations and its intricacies, and how this may actually open up some areas in terms of research needs for surveillance in some of the areas like Africa. Thinking about Kenya here, thinking about East Africa, thinking about places like Egypt. We think about persistence basically means within an animal, basically you have two categories of animal. One is, you may have, look at the diagram on the right, you basically have A and B. A is the top and B is the bottom one. A basically is non-vaccinated, so basically they are clinically susceptible, and you have primary infection, which basically is in the nasopharynx, nasopharyngeal epithelial tissues. And you have this dissemination into the whole body. So you have viremia, and then the lesions. This is what you see as kind of the lesions on the mucosa, some kind of lameness, fever, drop in milk yield and stuff. And then you have that particular animal recovering. So you have a convalescent one that has recovered. Occasionally, that may not really happen. And so you have the virus kind of being centered back onto the nasopharyngeal region. And basically it continues, it has a persistent infection, which is basically a carrier state. That's in a non-vaccinated individual. In a vaccinated individual, basically you have an element of protection. And so you have the primary infection, which is the epithelial cells, the precursor in the nasopharynx. And then you have this pneumotic subclinical infection, potentially passing on to from a dam to a calf. And then you still have this persistent infection. So you have this incubation, acute transitional phase, and then a persistent phase. Now, this persistence plays out in different levels in different populations, but basically you think about it in the context of different mechanisms. One of that is thinking about, okay, yes, we do have this persistence at the individual level, but at a population level, you can think about, okay, potentially this population keeps on being infected because it has a repeated exogenous seeding. Basically, you bring in an animal, or rather, there's some kind of a source from outside that comes in. When you think about this, then you think about like immune individuals, so those with the past infection or those who are vaccinated, basically does reduce the potential for the persistent within here. The herd management aspect of it, so herd turnover, you bring in more susceptible animals, infection of the whole herd, and things like vaccination. There's the other aspect of things like viral factors, the virulence and the fitness of the virus itself may also influence the persistence within at the individual level and at the population level. And then there's the role of carriers, which I'm going to explain in the next slide. When you think about some experiments have been done, especially in terms of experimental transmission from carriers to naive cattle. But there's no really good data that we have from the field, especially extrapolating that particular source of information to think about how that may lead to infection within a herd in the field setting. And then we have aspects of recombination and mutations. So when you think about recombination and mutation, in our work in Kenya in 2016, we were basically able to show that we basically sampled about 100 buffaloes and 100 cattle, and we recovered about 80% from the oropharyngeal fluids of the virus in the buffalo, and about similar number, 98% in the cattle. We then realized that we have co-infections of SAT1, SAT2 in about 6 buffaloes. The data I'm showing here is 3 or 4 buffaloes. And what you see here is one, I'm showing this just basically to talk about recombination and mutation and what that means, especially in terms of persistence within a population. What we were able to show here is when we use the normal molecular typing, we were able to classify them as shown here, 1, 2, 3, the numbers 1, 2, 3 on the left of the screen. The challenge was we tried to basically understand what that meant. And by the time we realized that we're actually having a biased assembly, so in other words, that classification is wrong by itself, partly because you're basically having a merge of 2 different sequences together. So we were able to basically use plaque, basically try to culture the virus. And when we did culture the virus and then we did next generation sequencing, we were able to show what you see there is basically recombination breakpoints that basically show that there's certain point mutations that lead to the, first of all, we were able to separate those into SAT1 and SAT2s. And then we have some of them that was a chimera, which was a 4 that we did not put in this particular analysis. But what we show from this particular diagram is we have recombination and mutations, especially when 2 different serotypes exist within an individual. Now, the question that we kept on asking ourselves is, will that lead to potentially emergence of a new variant that then is able to spread? Or how does that work within the population? The second aspect of it is thinking about host contact heterogeneities and differential transmission of FMD. Now, what I mean by host contact heterogeneities is you have more contact with your neighbors than those who live far away from you. That also happens with herds of livestock, where they're more in contact with their neighbors than those far away. But then if you think about a pastoralist population, then that particular principle may actually change a little bit. For instance, if you live within an area where there are common grazing grounds, or there's a common area where you can get a grazing ground, for instance, in the Masai Mara in Kenya, there's a lot of grazing inside the park, despite the fact it is banned, or around the park. Then you have people moving their livestock from far away into areas closer to the park, where they can access grass, they can access water. Now, that changes the contact patterns within that particular landscape, but also changes how the herds interact with the ecosystem. And so the persistent here basically is the high rate of contact vis-a-vis the differential rate of transmission of foot and mouth disease, combined with other landscape utilities like even markets. How does that at a population level change a highly evolving virus that's also highly infective? And so when you think about the scale here, the spatial scale, would you think about it in the context of the contact or in the context of the demographics of the animal vis-a-vis the immunodynamics? And so we start asking these questions in terms of some of the gaps here are how does the movement interaction shape the spatial scale of persistence, the scale at the population level. And you can still bring this back to the previous slide where at the individual level, we know what may happen. But then the question is, how does that play around with increased contact, with shared resources at a population level? And so we still think about this, especially in areas that has transboundary movement or even movement across large scales. And we start thinking about the lack of data, especially in expounding some of these and how that may lead to persistence of and maintenance actually of the virus within these populations. A good example here is from our paper in, excuse me, I think it's 2020, where we were looking at spatially differential herds. And basically what we have here is data from buffalo and cattle. Buffalo is basically the tips labeled in blue, and the cattle are the tips in black. If you look at SAT1, which is the left side of this particular diagram, the 2 boxes, which is basically a red and I think a green, basically show that even within Olpejeta Conservancy, which is a homogeneous conservancy, livestock and wildlife move from across one end to the other. The only difference is we have a river in between, which is the river Ewasoniel. And so this river divides the sanctuary into 2, but it's possible. Animals can cross across and stuff. But we see that even within that small area, we have a west. So what we have as W is west and E is east. So we have herds that we sampled on the east side and herds that we sampled on the west side. And you see that there's actually some kind of a geographical substructuring based on the river just by itself. And so that begs the question when you start thinking about the potential for these variants or the potential for a scale, a special scale within which FMD can transmit. How do we incorporate that? And that's some of the questions I'm kind of putting in, and this is where I narrowed it down to that. The same data can be quantified differently. So basically by measuring a phylogenetic distance, and this basically means the pairwise distance, the pairwise phylogenetic distance between the herds. And so we're using a community ecology principle here, where the null hypothesis is there's no phylogenetic clustering. And so what you see on the right is basically different herds, and we're trying to measure the phylogenetic distance between them. And what you see as a star basically means statistical significant at the 95%. And so we see very clearly the same, what we showed in the previous slide, that there are actually differences in terms of the structure, which basically shows that there is no transmission at a local spatial scale between these different herds. Now, how does that happen, especially in the context of they share resources, including water, including grass, or rather, including forage, but also they share space, and sometimes they interact, and especially in things like buffaloes, we know they exist in herds of 100 to 200, and sometimes they have this fusion where they come together and the herd breaks apart. Now, that begs the question in terms of understanding how this may actually be extrapolated to some of these areas that are potentially like a pastoralist area, or some of these arid and semi-arid lands. The other side of it is basically trying to understand persistence, is basically thinking about how emerging variants vary with herd dynamics. And so here I'm showing at a farm level, where you have the previous diagram that I used, we have a vaccinated and non-vaccinated, and a vaccinated and non-vaccinated individual animal in a farm. And so you have all these steps where you have a carrier species, a carrier status within the particular farm. And so you start thinking about, okay, how does herd demographics, let's say age structure, let's say bringing in more animals, change how, or rather impact how the potential for the formation, let me paraphrase that. How does herd demographics and turnover basically influence transmission or persistence of the novel antigenic variants? Here we're assuming that with the existence of a carrier status, there's a potential for formation of new variants. And then you think about kind of when you bring in more animals, let's say you've bought some animals, or you have another herd somewhere, and so you bring them to mix them in here. The question then becomes, are those particular individuals susceptible individuals? Now, if they're susceptible individuals, then we kind of ask the question here, which is a data gap here now, which is the presence of a susceptible population within the herd more critical than just the emergence of new variants? So in other words, can persistence just exist even with carriers alone, or with just the introduction of new animals within the herd, which basically form a new pool within which the virus can affect? And so the question here is, if those two scenarios are mutually exclusive, then what is the immunological picture that you see, and what is the epidemiological picture that you see? Those are some of the questions that I'm trying to ask here and basically drive the conversation around that. Now, in the same thought process here, we think about what happens at a community level. So here was a farm level, at an individual farm, one farm.
Yeah,
now,
George, you may need to summarize.
Okay. Yeah, at an individual level. Now, here's a community level where there's interaction between two different farms. There's interaction with the shared resource. There's interaction with the community, basically other wildlife. So here I'm showing buffaloes. And so we start thinking about the potential antigenic changes in multiple farms and communities, including things like markets. And then if there's an emergence of a new variant, how does it affect other farms and the shared resources, and what kind of an epidemiological picture do we see here? And then the last part of this is basically thinking about the alternative surveillance and control pathways, and basically research gaps here include, how do we identify areas within which there's congregation, and how do we apply things like network-based surveillance and control strategies? Basically, what I'm showing here is, on the left is basically a study from my PhD, which basically looked at animal movement within the Masai Mara environment. Adjustment is dry season grazing, which basically shows you where they go during the dry season. What you're seeing as big circles are basically villages that are utilized more. And the biggest one is the Masai Mara National Reserve. And here on the right, I'm showing basically the fact that if you actually use these villages and think about the villages that are being used more, and you try to do vaccination, that's what I mean by village removed. Using adjustment, you actually reduce infection much more faster. And so a targeted approach to surveillance and control may actually be better. So in conclusion, basically what I'm talking about here is, based on the research need, is if we try to localize surveillance and control efforts, we may actually have better outcomes at the national, regional, and the global levels. I'd like to thank the Foot and Mouth Disease Lab, Dr. Sangula, Kabugi, Kenya Wildlife Service, which now has 2 parts, Wildlife Research and Training Institute, Dr. Akuya, Ndere, Vincent Obanda, the College of Veterinary Medicine, Kimberly Van der Waal, and the USDA ARS, Jonathan Arts, Palinsky, and the team there. And thank you so much for listening to me.
Yeah, thank you, George. And yeah, that was very insightful with a lot of lessons to take home. Yeah, I think let's really rush to the next presenter, who's called Lesuenyo Motapele. Dr. Motapele Lesuenyo joined OIE in 2014 and is the OIE representative for Southern Africa. He previously worked for Department of Veterinary Services in Botswana, where he was appointed Director of Vet Services and OIE delegate for Botswana in 2008. In 2019, he became a Deputy Permanent Secretary in the Ministry until joining the OIE in 2014. He was a member of various technical committees of international organizations such as OIE, FAO, AU, IPA, SADC, etc. All this culminated with an award of meritorious honor by OIE. So let's welcome Let's to give his presentation. Thank you so much.
Thank you very much, Mr. Chairman. Good morning, good afternoon, good evening, colleagues. As we heard, my name is Motapele Lesuenyo. I am presenting here on behalf of the GFRA working group, which I'll explain later what it is. And I really have to thank the organizers for giving us the opportunities, and I recognize all of you participants, colleagues, and other members of the working group which are in attendance here. So basically, my presentation layout is as follows. I'll go through the overview of the progressive control pathway for FMD, the training needs, available opportunities, and give an example, and also provide sources of additional information. So starting, for a start, the GFTAS, or Global Framework for Progressive Control of Transboundary Animal Disease, GFTAS in short, was formed in 2004 as a joint initiative of FAO and OIE. The two organizations basically combining their strengths to achieve agreed common objective. A very noble initiative indeed. Under the GFTAD framework, FMD, or foot and mouth disease, is a priority disease. There are others such as FMD, SF, and so forth. But FMD is a priority disease and has a specific working group on it, which is comprised of FAO with technical support from the EuFMD. This is the group that I'm representing, and at the end, you see the members of the group. Some of them are in attendance here. So the FMD working group developed the PCP, specifically FAO and EuFMD, in 2008, which was aimed to progressively reduce the impact and burden of foot and mouth under endemic settings, such as we have in Africa. The PCP approach was adopted by FAO and OIE as a working tool in the design of FMD control and regional control programs. So you can see it's a very, very handy tool. It consists of stages that progressively increase the level of foot and mouth control through identifying and addressing risk of FMD introduction and spread. So because we are talking of endemic settings, risk is the key thing. There is a key thing that we'll talk more about. It was revised, that is the PCP, in 2018, and now consists of two distinct parts, which is the GFTAS, the original GFTAS part, in stages 0 to 3, stage 3, as well as the OIE part, which is stage 4 and above. But the principles are risk-based, as I've said, non-prescriptive, and progression is based on evidence. It has to be very, very evidence-based. So there's actually a whole document on the PCP, which you can access at your own time. So a very quick overview. Those are the stages, the 3 stages. Stage 0 basically meaning nothing is happening in that country or in that zone. Whereas stage 1 is when the country starts to identify the risks, and the risk may be related to high probability of disease entry or spread. So it's still a very dangerous area. And normally to move out of stage 1, you need to have something called to prepare a risk assessment plan. Once again, you are trying to contextualize and document the risk situation of the country. It is very important to progress from stage 0 to stage 1, to be in stage 1, that is. And then to move to stage 2, you need to have a risk-based strategic plan. Now, at this stage, the risk is starting to be controlled and the impact is reduced. And resources may be targeted on a particular sector, say the dairy sector or the breeding sectors, whatever the sector could be according to the country's priorities. And the countries may remain in stage 2 for some time, depending on the disease situation, the priorities and the resources. And stage 3 is when now the country is becoming more advanced. There's progressive elimination. The country is going towards elimination, the OI stages. And there is a national official control program. So they need to rapidly detect and respond to all outbreaks in domestic animals. So the country is becoming more organized here. I think it's very important to remember this: the risk assessment plan, the risk-based strategic plan, the official control program, because all these are the requirements to progress from one stage to the other. And this is where normally resources are required and a lot of help is required. And we are talking in the context of triple P, that the private sector, not just the government sector which can help, the private sector can help in addressing this, because these are very, very important plans. So just continue with the overview of the PCP. The country requests are evaluated by the GFTAS working group and approved by the regional advisory group, which is a selected member of selected members from the member states at a regional level, who are responsible for approving whatever the working group would have determined, whether a country progresses from one stage to the other, and so forth. The RAG, as it is called, also identifies the training needs. So they are the people on the ground, they know what is required. They can be feeding on the working group on what is required. And occasionally there are these regional roadmap meetings and targeted workshops. And these are areas where now there is a whole exchange of information between the member countries and the working group. So that is the overview of the PCP. Just to mention that the stages 4 and beyond, these are the OIE-free statuses, normally starting with the endorsed control program that is moving from here now upwards there, and then having FMD free with vaccination and having FMD free without vaccination, and also talking about maintenance. All these stages, all of them, even the PCP stages, it's very, very important to have a program of maintaining the status, because if you don't maintain the status, you will relapse to a lower stage. So now focusing more on the triple P, which is the public-private partnerships. The OIE has actually an effort, like it was said, there's a whole website dedicated to triple P. And we start by defining that the triple P is a joint approach in which the public and the private sector agree responsibilities and share resources and risks to achieve common objectives that deliver benefits in a sustainable manner. Benefit from the private sector can be direct or as a corporate social responsibility. So you can see it's a win-win. There has to be a benefit for the public sector, a benefit for the private sector. And FMD control can be one of where this PP can work. Of course, we know that foot and mouth control is largely, the regulation is largely done by the public sector, but the private sector can contribute to this financially and in kind by availing things like experts and so forth and so forth. That is according to the P platform. So the PCP is about FMD control by identifying, addressing the risks of introducing FMD, as I've said. In other words, managing the risk to progressively reduce the impact. Therefore, the training, all the needs that we are going to talk about here at each PCP stage are generally for the merits of controlling foot and mouth disease. And it's all about managing risks. Risk is the most important thing. All the different aspects of risk analysis as I've outlined there. And the private sector can have a role to play. First of all, informal training by mobilizing grassroots level on FMD and its risks and how to control it, as well as maybe sponsoring formal training at a high level so that there is a pool of expertise on FMD control at country level. So I'm starting to try and relate how this control of foot and mouth is not just for government only, but the private sector can play a role as well at different levels. The socio-economic impact assessment. The private sector can provide resources for a cost-benefit analysis, which will, among others, guide the investment and effort. And this will be used to guide progression along the PCP stage. So basically, as you see, the risk assessment plan, the risk-based plan, all those things, they have different economic benefits that can be analyzed and can guide the private sector as well as the public sector in how they do investment, looking so that they can get return on their investments. Laboratory diagnostics and surveillance, very, very important, as we said, we had in previous presentations. And the private sector can finance laboratory equipment as well as their maintenance, because this is always a challenge in governments, and provide resources for regular FMD surveillance, especially in higher PCP stages, which are more demanding. Basically, you'll find that if you go through those stages, there's going to be more increased evidence-based surveillance and diagnostics, and the private sector can help there as well. Post-vaccination monitoring, the public and private sector can collaborate in availing resources and doing post-vaccination monitoring. With DVS, vaccine manufacturers can, of course, with the help of DVS, vaccine manufacturers can do finance PVM for them to appreciate performance of their vaccine in the field and guide improvements accordingly. I think we heard that from BVI yesterday, that this is very, very important to see that the vaccine is out there in the field, is doing what you intended to do. The concept of safe trade, which is what we do. We do all this animal health to have safe trade. And the private sector can adopt best practices to enable production of disease-free commodities. I'm talking about farmers here and the industry in general, that production can be done in such a way that you are producing disease-free commodities through the entire PCP process, whichever stage, if it can be a way of doing things by the farmers and the industry. An additional private sector can assist in the implementation of international standards to ensure safe trade of animals and their product. In other words, the private sector should not wait for government to be the one imposing all these restrictions on. They should adopt a culture of best practices of producing in a sound manner. And the overall, that will have an overall benefit of growth of the livestock sector and increased contribution to the economy. Another training needs that we can identify in the triple T perspectives, monitoring and evaluation of control of foot and mouth. The private sector can assist with doing monitoring evaluation of an FMD control programs or plans, especially between progressions. Like we mentioned, there are all those requirements, the risk assessment plan, the risk-based strategic plan, and so forth between the different stages. It is very, very important to always pause and evaluate, monitor and evaluate the performance before you can move to the next stage. So the private sector, with all its resources and its knowledge, can actually assist in that. And designing those programs themselves, those plans themselves, as I've mentioned, the private sector can give a hand on that, because those are not just documents which are written, they're actually well-researched, evidence-based activities, work plans that are out there. And the vaccines and vaccine strategy use, timely vaccine availability is always a challenge in the public sector. The private sector can assist and improve foot and mouth control and ultimately the overall benefit to the country. And lastly, advocacy and communication. This is not exhaustive, but we are saying that in some countries where the farmers association, farmers unions are very powerful, they can lobby government to give more resources for FMD control. So currently, opportunities which are available is that virtual learning courses which are available from the EuFMD. The OIE also offers virtual learning courses from the OIE learning platform, and these are open access and available most of the time. Others are periodically offered. The training needs may be specific to FMD or to apply to other diseases as well. These are opportunities which exist, which can be used. And also at country level, the working group has this PCP support officers or PSO system, which is available to provide tailored assistance for countries to progress along the PCP. So this is an opportunity that can be utilized. And once more, there's good opportunity at different levels for the PT to survive. I tried to look for a good example where I can show what I've been talking about. Unfortunately, I don't get it. I could not get any validated one for PCP, but this is a very good example of triple P working for FMD control, a case in point in Namibia. You can also see it on the OIE website on triple P. Basically, there's a very good collaboration between the Meat Board of Namibia, which is a private sector party, and the Directorate of Veterinary Services, which is a public service partner, bound by a memorandum of understanding. So this is a partnership that is very, very serious. And the triple could quickly mobilize funds to assist DVS to immediately set up control measures. And those are the activities here that this partnership does. They have something like a secretariat, which is called the Animal Health Consultative Forum. And it does all these are the activities that are done. And it is, you see, it's all that I've been talking about: awareness, appointment of experts or availability of experts, coordinating veterinarians, provisions of rations to temporary staff. You see how the private sector can come to the party and help government in a lot of ways, even providing basic things like rations to temporary staff, many roadblocks in an emergency. So in this arrangement, you have the public and the private, which is here is the Namibia Veterinary Services, and this side is the Meat Board of Namibia. And these are the different activities together working for a common purpose. And the benefits are improved animal health, regulation, maintenance of export markets, and the impact that has been seen there is in the economy, which is very much for the public, where the livestock sector contributes more to the GDP, and the population, especially a population that is dependent on livestock, maintenance of trade, that is very, very important benefit at public level. And also at public level, the trust, where the private partner can inform, but also advise DVS on implementation and compliance to market requirements. It's a partnership. And of course, with the private, very much business-oriented, maintenance of existing and emerging new lucrative markets, value livestock, and so on and so on. So basically, you can see there are advantages for the public and for the private. It's a win-win situation. So even though this is not specifically on PCP, but it's still on FMD control. And I think there's an opportunity that any country can apply the same kind of concept at a level of the PCP. So just going towards the end, these are the available informations that you can look at your own time. These are mostly OIE in this page, the handbook, we have the online courses, the training portal. We even have this OIE database, which is where I got this example from. But there's also from the EuFMD and the FAO's, the EuFMD and the FAO site. So there's a lot of information on triple P. And in conclusion, I just want to say that one of the major constraints in Africa to controlling animal diseases, especially foot and mouth disease, is availability of resources. But so many times the private sector has resources. And sometimes we feel that maybe the public sector is not too open to receiving the private sector. I think time has come that the public sector has to look upon the private sector, and the private sector should be willing to come to the table and share resources, share expertise, such that they both work together for the common good of the country. It's a very good opportunity, especially in the ever-decreasing resources allocated for animal disease control. And finally, I just want to acknowledge the contributors to these presentations. These are members of the working group on FMD. You can see all of them there from the FAO side, EuFMD side, and the OIE side. I thank you, Mr. Chairman.
Thanks so much, Lex. That was very, very interesting, and it provides a very broad background on how to approach PCP and the opportunities available. So at this moment, allow me to proceed to the second last session, which will be a round table. It will be facilitated by Theodore Knight-Jones and Sylvia Baruka. Allow me to also acknowledge the CVO of FAO, Keith Thompson, who is online. And of course, together with the president of GFRA, they will be facilitating the last session. But for purposes of this session, the round table, I call upon, let me first of all introduce Theodore Knight-Jones. Theo started his career as a clinical vet in the UK and South Africa. He then specialized in veterinary epidemiology and public health work in Europe, Africa, and Central Asia. He now works as a principal scientist for the International Livestock Research Institute, focusing on East Africa, where he leads work on herd health, including research on improved delivery of animal health services through public-private partnerships. He also leads major research projects on food safety and One Health. His research of interest are epidemiology, economics, and policy for transboundary animal diseases and zoonosis. At this moment, allow me to thank you and request Sylvia and Theo to take us through. The questions in the chat box, some of them have been answered, but the rest will be elaborated on. I wish you success. Thank you so much.
Thank you very much. And I think we've got about 20 minutes. So yes, thank you. Sylvia will help with a roundtable discussion with me, but she's also a presenter, so you have two hats. So if you like, I'll post the first question, and it came up during one talk, but I think it is relevant for a few different speakers. So we talked here quite a bit about knowledge gaps and research knowledge gaps, but to what extent do you think it's, and I'll direct this question first at George, and if all the speakers can put their camera on, let me know who's online. Yes, George, to what extent do you think it is these research knowledge gaps that are holding back FMD control in the situations you described, or in Africa in general?
Thanks. I can't say they're the only ones holding back, but I think the aspect of FMD control, especially in African context, is a mix of multiple factors that we may need to dig out one after the other, or sometimes kind of look at it collectively. So a good example is when you think about research gaps here, I'll give an example of the 2001 UK outbreak was caused by serotype O. In that context, especially in a country that we may consider as clean, then it means its control is basically, in a way, a linear thought process of, okay, what are the areas that are important, which are the herds affected, which are the farms affected. In the African context, especially in East Africa, there's multiple aspects, combination of different things, including low vaccination rates, movement of animals, unregulated movement of animals, lack of good quarantine policies, especially implementation, things like sometimes mismatch of the vaccines, and then the other aspect is the carriers and stuff. So I think in terms of holding back, I think is a multitude of factors, and all these form into some kind of different facets that could be tiered to be able to basically be able to control the disease, but not necessarily eradicate it completely.
Okay, thank you. And Sylvia, you can just add when you have a question. I mean, otherwise, I'll pass that to Lech, but to say that from your talk, Dr. Lechwania, what are the key issues for getting the public-private partnerships going and making them sustainable? And then after you, maybe I'd pass that on to Sylvia, because she also spoke about these public-private partnerships.
Yeah, thanks, Theo. I think one of the things that I talked about is maybe we need mindset change, whereby the public sector is a bit more open to private sector, especially for the control of foot and mouth. Traditionally, in the African setting, government always take the lead role and does everything, implements, regulate, and so on. It's basically doing everything. But the private sector has to be more involved. So government has to change its perception and be more receptive to the private sector. Now, the private sector itself, it needs to be a bit more organized, because it's very, very difficult for the government to be talking to individual farmers or individual few numbers of private sectors. We see in countries where they have very good association, farmer associations or industry association. It doesn't have to be just farmers. or farmers union, that these people they have a lot of resources, they have a lot of knowledge, and when they go to the discussions with government, they can actually end up with a win-win situation. So I think it's more of a mindset shift, both at the private sector level and the public sector level, so that we all work along that same way. And I think this to add to a question that is in the chat also. Someone was asking, how do you identify the willing private sector? I think once again, the private sector is better identified when they are organized, when they are into associations and unions, especially when they even now have umbrella associations, which comprises of different regions of the country. So, you know, the private sector needs to be more organized to be seen, and the public sector has to be more looking outward to see the private sector out there. Thank you.
Thank you. Sylvia, over to you.
Yeah, thank you very much. I would like to add by saying public-private partnerships can be viable, and they can be a vehicle that would help us to be able to improve not only animal health service delivery in general, but also improve animal disease surveillance. However, institutionally, there are weaknesses. I think it's not only on the private sector side, but even on the government side, I think there are also institutional weaknesses that need to be addressed to make PPPs an attractive vehicle that we can use, that we can work through. For instance, as much as George has just indicated, I do agree that to a great extent, our private sector seems not to be organized. However, on the other hand, I also note that on most of the time, the public sector doesn't seem to recognize, or maybe let me not use the word, okay, let me avoid using the word as not respect. Let me say, most of the time, public sector doesn't seem to recognize the private sector as a potential partner, that they can come together mutually with mutual respect and be able to enter into a commitment and agreement to work together. Most of the time, when private sector is invited on the table, it is either invited too late, or it is invited when most of the time the terms of reference are already drawn or defined. And most of the time, private sector, I think, doesn't get to feel that they're actually needed and they are valued and they are at an equal footing. So I do agree that we need to support the private sector, and the private sector players need to come together and strengthen themselves. But at the same time, I think we need to review some of our policies so that they can enable the public sector to actually recognize the potential and also the benefits of working with the private sector. And if we address those institutional weaknesses that touch the frameworks, the structures, and also as well as the policies, I think the rest of the aspects can be able to follow. That would be my contribution to that question. And I ask you to continue with the questions, please.
Thank you, no problem, Sylvia. Over to you, Keith. Thanks for joining us for this roundtable. Maybe you could add some thoughts on this. What some of these barriers, including delivery through public-private partnership, and how, and the knowledge gaps, and how many countries in Africa and elsewhere sort of get stuck at certain stages on the PCP, sort of going from 2 to 3, and what are the key barriers and how can they be overcome? Here we're just talking about FMD a lot, but now in your role now you look at many different diseases, and maybe that's one of the reflections as well, joining up of resources. Over to you, Keith.
Thank you, Theo. I wasn't thinking of coming in now, but that's an interesting question. Yeah, I mean, maybe what has prevented countries moving from the concept of control, which might be largely paid for by the private sector, or at least it might be just partial to some sectors, to stage 3, in which there's really a move to suppress and control virus circulation. I think there's really sort of 2 ones, but they mostly relate to the appetite of the private sector producers, livestock producers, processors, exporters, for moving to a higher level of control. I think if there is that strong and strength of associations representing the from the smallholders up to the larger commercial sectors, together with a potential export policy, you know, that's what's driven in other countries. Now, maybe what's missing is the kind of co-development of policy that in this sense, because it costs a huge amount to move from PCP stage 2 to really effectively do stage 3. It needs a lot of buy-in from public and private for that leap. One side trying to do it isn't going to work. It needs to be done together. So in certain regions more of the world, I would say, maybe the kind of meetings in which policy is decided would be have a much stronger representation of the private sector in them than we see in most countries which are in stages 1 or stage 2. So I think that some of that will come with a greater voice being given to cooperatives, associations, sectoral interests to do it.
Thank you, Keith. Going back to you, Sylvia. I mean, from your experience in PPP, how do you overcome this sustainability? You know, often farmers that are playing a major role in FMD transmission have very few resources available. They have a lot of cows there, some of them, which might be worth a lot of money, but there's very little money available to pay for an ongoing service through the private sector, or a reluctance to pay because of previously it's been subsidized, or reluctance to take preventative measures. You know, you deal with the crisis when it happens. How do you overcome that situation to have a sustainable private sector playing a role in this?
Well, thank you so much. I think we can achieve a sustainable private sector in animal health service delivery, more so by the veterinary paraprofessionals, but also by the veterinarians, if we come to an understanding that as key stakeholders, if we looked at the livestock sector as a value chain and look at it as key stakeholders who have a role to play, but also we take benefits from this sector, and that would require a sort of a mindset shift. When a farmer invites me, I don't go and look at this farmer and his animals as if they need my service and all I need is money from them. The service that I give them, for instance, if I've been invited to treat, I should be able to treat, but also provide advice to this farmer, because most of these diseases could be prevented just by observing very good farm biosecurity. And sometimes, what I always tell my colleagues, ourselves as animal health workers, sometimes we are defaulters ourselves. We don't seem to respect the biosecurity rules, especially in our resource-constrained environments. But if we promoted that, and we actually encourage the farmers to observe biosecurity, knowing that when these animals take long to get sick, they are more healthy, their productivity is higher, the farmer earns more, and the farmer is able to save much more after providing for the household and for their livelihoods, then this farmer's capacity will improve to be able to pay for my service. So there is something that needs to happen at all levels, from the farm level, the farmer's point of view, to the primary animal care service providers like the community animal health workers and veterinary paraprofessionals, but also for us, the veterinarians who supervise the entire sector. All of us need to become more keen. We need to become more keen in the way we work, and the way we serve, and the way we relate. And also, of course, improve our communications and feedback. If we do that, I think we'll move towards achieving sustainability. That's what I want to say, and I want to emphasize biosecurity and then good communication, good stakeholder engagement, and stakeholder partnerships and relationship management. If we're able to achieve that, I think there are a lot of prospects and potential within this sector. I thank you.
Thank you. Dr. Lechwini, perhaps you would like to reflect a bit on specifically this bit of is who will pay for this private sector aspect of these partnerships to make it sustainable?
Yeah, I think the industry, if you take the whole value chain, there are a lot of players there, and between themselves, they can raise quite a significant amount of resources. It's just that if they are doing it piecemeal because they are not structured, then it looks like they don't have resources. But generally speaking, if you take the whole value chain, and there's a good appreciation of the value chain by the public sector, they can be mobilized to provide those resources. And I really want to underscore what Sylvia was saying regarding value chains, because I think so many times you look at livestock production, maybe we just think of the veterinary services and the farmers, but there's a whole lot of other people there. Dealers there, feed millers there, exporters, abattoirs, feed processors, and so forth and so forth. All these people, they have a role to play. And so many times they do have resources, and they are willing to put those resources forward, provided there is a justification and they can see the benefit on it. And like I said in my presentation, benefit can be direct financial, or it could be in a way of visibility as like corporate social responsibility, where they want to be seen to be doing something to the community. It's a big company, they are doing something to the community. There's a better appreciation of their company, and there are better returns in their company. So for me, like I said, I think we need a mindset change, both sides, both on the public side, the private side, and realize that we are in this together and we can do it together. And resources will naturally come with this kind of partnerships. Thank you.
Thank you. Over to George. You spoke quite a lot about different and developing surveillance methods and systems. How can we factor these in a sustainable way that can be funded and also linked into active responses to actually affect the burden of FMD?
Yeah, thank you. I really like what Sylvia and Let's are kind of putting across, and I think this is an area that could be explored. To give an example, for instance, in Kenya, we have dairy circles, which basically started from the ground upwards and basically led into the institution of certain, potentially certain standards, including feeds, vaccination, and taking care. And I think when we think about the FMD and its occurrence, especially in East Africa, we are looking at more of these extensive farming areas, which are basically arid and semi-arid regions. And the challenge has always been access to veterinary services, especially from the government perspective. And the implementation of some of, especially the PPPs, may actually come in handy if there are incentives for people, agrovet, these are basically the shops selling medicines, vaccines, providing services to the farmers, if there are actually incentives for them to set up in place, either individually or some kind of a wheel spoke method, which basically allows for an individual in an area. The sustainability part of it is tied not just in disease control, but also in the productivity of these ventures. The productivity basically stems from the added advantage of spending one extra dollar to vaccinate an animal, or spending $40 to vaccinate a group of animals. What does that translate into in terms of the productivity of the farm, but also in terms of the sales? And I think the challenge we are having here is there's literally no cost effectiveness in the cost benefit analysis, and more or less the benefit in terms of for every cost incurred by the farmer, what will it actually mean? Partly because of the low mortality rates that FMD causes, but also because most of these particular farms do not really see the potential drop in yields, partly because they're not, for instance, high dairy producing areas, they're basically beef and very low milk producing areas. And so I think tiering these in the sense of trying to start showing that there's potential to gain by having disease control may lead to willingness of the farmers paying for this particular vaccine, but also willingness of the private sector taking up some of these challenges, either through farmer federation or the formation of things like co-ops that allows them to actually either get good breeds, get better stock, and also translates into sales.
Thank you. And I think we've got time for just one very quick question. So I'll ask Dr. Lechwenio again about how these PPP, we're talking about surveillance and delivery aspect, but we're often just here, we're just talking about FMD. But how do we factor in how these services are really multi-disease services? How do you make sure that's captured? Because that just brings a lot more value to anything that you would do.
Yeah, I think you are right. You are very right, Theo. These are multi-disease interventions that can be used that way. I think one way of doing that is maybe having a good contingency plan or good contingency plans, so that during peacetime, when there are no outbreaks, the different value chain actors can be brought together and formulate a contingency plan that will describe and explain in detail the role of every partner or every sector during an emergency. That way, everybody knows exactly what they have to do when there is an outbreak of a transboundary animal disease. And contingency plans, you can use FMD as a model, but that will also apply to PPR, to CBTT, to African swine fever, and so forth, because these are the principles, especially the epidemiological principles, and also the monitoring and evaluation, and so forth and so forth. So I think, to answer your question in short, I think we need to emphasize on the development of contingency plans, done well in time, all inclusive, everybody having a role to play, and everybody knowing what their role is going to be in an outbreak situation. And this will not just be the public sector. It will not just be the veterinary services. It will also be the other public sector that are not veterinary services. For example, if you want to use the army or the soldiers to help in the patrols, they need to come on board. Or we have to ask to engage the transport organization responsible for government fleets. They have to come into the party at that stage so that they know and they internalize their role well in advance, as well as the private sector, the abattoirs, the breeders, the dairy farmers, and so forth and so forth. They also have to know exactly how an outbreak of a transboundary animal disease will affect their sector, and therefore what role they can contribute, and it be documented well in advance so that should it happen, everybody knows exactly what to do. Thank you.
Thank you. Chair, do you want me to carry on the discussion for a minute longer, or wind it up? I can, if you like, if we want another minute or a couple of minutes.
Yeah, I think. Yeah, I think you can quickly wrap up so that we go into the closure. So okay. I think you carry on and wrap up. Thank you so much.
Okay. Well, I think it has two more minutes then, and so I'll hand to Sylvia just to reflect a bit on you doing this PPP project, but within an FMD background. But so, how did you deal with this issue of incorporating other diseases within your work and evaluations? Just 2 minutes. Thank you.
Okay, thank you so much. Yes, as you have correctly put it, Uganda is still endemic for foot and mouth disease, but also other fast diseases important. For instance, PPR, Rift Valley fever, and a number of other transboundary animal diseases. Now, what the project is doing, the scope of this project is addressing competencies, capacity building for the veterinary professionals, and then also providing, supporting them to create a business environment that can help them to establish and operate sustainable businesses in animal health service delivery. So when we right now, the project is not yet 6 months since since the inception. So in the first phase, we have been developing the VPP competence framework. We've been working with a number of VPPs, identifying their needs based on the needs of the farmers whom they serve, and based on the challenges that they face, and also based on the limitations that they have in their current levels of knowledge. So for as long as the VPPs identify particular aspects, for instance, in some areas, poultry are more important, and therefore they're talking about poultry vaccines, poultry diseases such as Newcastle disease. In some areas, you find that pig farming is important, especially in the peri-urban areas. So we are working with the farmers with all the important animal species in the different project areas. Although we have priority on FMD and other fast diseases, but we don't leave out any important challenges that the farmers identify and the VPPs identify. So we are working with them, and the competence framework has been developed based on those needs. And the trainings will also be provided to address the actual needs that have been identified through a PRA process by a team of consultants and through group discussions, through key informant interviews. So we do believe that the trainings will be responsive and will be meeting the actual needs that are felt by the VPPs and also by the farmers, because we have gone out to the ground to actually engage with them and also look at the reality of what is actually needed. So there is no disease that will be ignored. There is no animal species that will be ignored under this project in the 3 years.
Well, thanks. I think that's a very good take-home message to sort of round up our round table of the importance of listening to the needs of the end users and providing what has a demand, what is demanded by these end users. So that I'll thank all the speakers and for their participation. I'm in the round table and hand back over to the chair. Thank you.
Thank you so much.
Yes, thank you very much, colleagues, especially the panelists of today and even those ones of yesterday. Allow me also thank the participants of yesterday. That is the over 180 participants online. And even today, I can see the number is 149 participants. This shows the interest in FMD in endemic settings, particularly in Africa. My observation from yesterday's talk, especially from Don King, and one of the key issues that I've taken out is that we need a fit-for-purpose FMD vaccines. This one, we should take it seriously. Then on personal note, as somebody really who is interested in FMD all over my PhD work and even other works, even after my doctorate, is that we need more surveillance to be carried out on the African continent. Look at the Democratic Republic of Congo and go to West Africa and Southern Africa. We have got a little data that we need to build up that database. Let us do the needful, especially the scientists across the world, and we can involve actually the PhD students or MSc students such that we can build a very good database as an addition of whatever we are doing across the world. Also, I will welcome you to the Global Foot and Mouth Disease Research Alliance 2020-2023 next year that will be held in Uganda. I also thank the members and organizers, that is Ariandra Capozzo, Will Navoslo, Nadia Rimich, Fabrizio Rosso, Alexandra Alivit, Rodvika Nella, Alice Namatov, Benedicto Vyamukama, and Nagendra Singanora. So thank you very much, colleagues and comrades, for all the wonderful presentations that we have had right away from across the world. And Giorgio Mondi, we woke you up very early. I thank you very much for attending. On that note, allow me call Mr. Keith Sampson, the Chief Veterinary Officer. We are privileged to have you as the leader of the Animal Health Program and the Director of the Joint Center for Zoonosis and MR. We know that Keith has worked on disease ecology and the interaction of wildlife, domestic, and environment for more than 30 years. And from the initial studies on how disease can affect ecosystem balances. Through his PhD studies on African swine fever and wildlife and domestic hosts in Southern Africa, he has got 12 years as a research team leader from the University of Edinburgh and DFID Kenya, and more than 17 years of management and epizootic diseases, especially FMD, including in the Wildlife Domestic Interface, and also as secretary to the EuFMD Commission based in FAO. Since June 2020, Mr. Keith Sampson has been the Chief Veterinary Officer for FAO, and since July, the Director of FAO's Center for Zoonosis and MR, and the lead of FAO's animal health services. Mr. Keith Sampson, please, you're most welcome for these closing remarks as we end our online live streaming on this FMD in endemic setting. Thank you very much.
Thank you very much. Thank you very much, Chair, for this. Welcome to me. And I'm really thrilled to see the number of participants from the range of countries, range of backgrounds, range of institutions meeting. I think, Frank, if you may, last time we met was at a GFRA meeting. And we talked there about how do we improve the networking of the academics, the public and private on FMD in Africa. And we exchanged ideas, and a whole group formed around us who wanted to debate and discuss this and how to do it. I think today is, and last couple of days is a bit of an example of this. You know, we need the oxygen coming from all these different voices. We need the academics from around the continent to have their own work and their PhD students, their masters, their own undergraduates thinking about these things that we've discussed today, because they are the reality. And we do need these things to be discussed. So really like to thank GFRA, EuFMD for organizing this. I hope this is one of many such kind of dialogues that we need in this area. Some of the issues are broader than FMD, many of them are, but FMD is a good example of where things really need to come together if there's to be any action and any progress. I remember from the time I worked 25 years ago in Kenya, that when we did a survey of the agro-vet stores, and we asked people coming out, what is it that you bought? What is it you would have liked to have bought if it was available? I was amazed, because I didn't work on FMD really that much at that time, when the number one thing they said they couldn't get was FMD vaccines. That probably has not changed in many areas for people who keep cattle. So we have a collective problem. How do we ensure that the means to control the risks of FMD are felt, they're tangible at the level of livestock producers? Is it entirely the public responsibility to make them available when we need the private sector to be producing quality vaccines, wanting to import them, managing to have them registered, have them pass through the value chain, and the person delivering them to make money at the same time? All of that has to work, and the vaccine has to work. Otherwise, there's no demand in the end. Now, we know a number of those things which are barriers. Some of them are scientific information. Much of them are about the showing that there is a market and that there is a system to keep all those parts working together. So I really welcome these discussions. There's some great presentations I can see have taken place. There's some super initiatives which are working on this, which will be, I hope, showing tangible results, such as the FMD vaccine challenge in the coming years. I would really encourage that we manage to keep these dialogues going. I would encourage you as academics as well to play your role in networking and to ensure that we keep meeting. And I'm sure that colleagues in GFRA and EuFMD would be happy to technically support further dialogues, further discussions in this point. At a meeting last week, someone made the analogy that if we take an egg and we break it from the outside, usually we destroy the life that's in it or the potential for life. But if an egg is broken from the inside, then usually that means there's new life. So thinking about that, the people in this meeting are really the life inside the egg. But maybe sometimes we don't know how thick the shell of the egg is. We don't know how big the problem is. And we need help from outside as well. Together we can share what is relevant to break those barriers and then to bring new life and new ways forward. I'm convinced that much of the talent needed to identify the problems are in this call. Maybe there are others we need to help work on those solutions that are needed in order that the great ideas that you've identified
will continue to be addressed in the right way. So with that, I'd say keep going, keep meeting, and we're listening, and we want to help and do our part to reduce those barriers to access to effective means to control FMD at the farm level, at the country level, and ultimately at the continental level. Thank you.
Thank you very much, Keith, and very good closing remarks. So colleagues all around the world, thank you very much. And lastly, really, wish you a good day wherever you are, and let's keep in touch and we keep this network really together, because we are the people really across the world to do our best. Thank you very much. Okay, bye-bye.
Bye-bye.
Bye, thank you.
Bye. Bye.